The overall goal of the following experiment is to investigate how transfected fluorescent proteins are distributed and move in the endoplasmic reticulum, and to assess the effect of expression on the ultrastructural architecture. To do this, cultured cells are transiently transfected with GFP-tagged tail-anchored ER-resident proteins. Following transfection, live cell confocal imaging is performed to assess fluorescence recovery after photobleaching, or FRAP, and fluorescence loss in photobleaching, or FLIP. Ultrastructural analysis is performed using transmission electron microscopy. The resulting data demonstrate that the expression of GFP-tagged B5 construct dramatically alters the organization and structure of the endoplasmic reticulum, inducing the formation of aggregates.
The method we demonstrate here can provide insight into basic cell biology processes such as protein and lipid mobility and trafficking. It can also be used to study responses to pharmacological treatments, to evaluate fluorescent protein oligomerization, or to study the role of pathogenic mutant proteins in the formation of intracellular aggregates of ER origin that may be relevant to their pathogenicity, such as in amyotrophic lateral sclerosis.
The plasmid used in this study consists of an enhanced version of GFP fused at its C-terminus to the tail region of the ER isoform of rat cytochrome B5 via a linker sequence. The tail region of B5 contains the entire sequence that remains membranous.